Hello dear students. Today our topic of deliberation is sanitizers and antibiotics used in the food. First, let's discuss the need of sanitizers and antibiotics. One of the major advances in human history was the ability to preserve food. It was the prerequisite to man settling down in one place instead of moving from place to place in the never-ending hunt for fresh food. The development of food preservation methods may be divided into a number of stages or eras depending upon the techniques used to maintain the foods in an edible state. Animals are treated routinely with antibiotics to prevent, treat, or control disease. Even under the best conditions of agricultural management, crowding and stress can lead to disease. The objective of cleaning and sanitizing food contact surfaces is to remove food, nutrients that bacteria need to grow, and to kill those bacteria that are present. It's important that the clean, sanitized equipment and surfaces drain dry and are stored dry so as to prevent bacteria growth. Similarly, the need for a simple, inexpensive method of reducing losses of fresh food due to bacterial spoilage prompted an investigation of the value of antibiotics. Antibiotics and other antimicrobial agents are used widely against pathogens for protection of human and animal health. Production of sufficient safe and nutritious animal-sourced foods for human consumption is necessary for the security of food supply. To ensure this production and animal well-being, proper management of livestock includes appropriate use of vaccines, parasiticides, good handling and housing practices, and appropriate nutrition. These practices can reduce but not eliminate disease incidence. Prudent use of antimicrobial agents remains necessary for prevention, control, and treatment of infectious disease in food animals. Dear students, let's now discuss sanitizers used in the food. To begin with, let's first see what sanitization and sanitizers are. Sanitization is defined as the process of reducing microbial load from the surfaces to the levels considered safe as far as public health is concerned. Sanitizers reduce but do not completely eliminate microorganisms from the inanimate environment. There are two types of sanitizers: non-food contact sanitizers and food contact sanitizers. Non-food contact sanitizers. These must demonstrate a 99.9% reduction against gram-positive, S. aureus, and gram-negative bacteria, Klebsiella pneumonia, or Enterobacter aerogenes over a parallel untreated control in less than or equal to 5 minutes. EPA policy does not allow tuberculocidal or fungicidal sanitizing claims. Food contact sanitizers. These must demonstrate a 99.999% reduction against gram-positive, S. aureus, and gram-negative Escherichia coli over a parallel control in 30 seconds. Though the EPA requires effectiveness to be demonstrated in 30 seconds, existing policy limits the contact time stated on the label to 60 seconds. Next, sanitization in food processing. Sanitizing, that is the reduction in the number of microorganisms, is required in food plant operations in which wet surfaces provide favorable conditions for the growth of organisms. In many situations, thorough cleaning will provide adequate microbial control simply by physically removing the microorganisms or by removing nutrients which they require for growth. On the other hand, the rapid growth rates of bacteria in some foods require that equipment surfaces be almost sterile during operation in order to produce a wholesome finished product. Let's move on to the properties of sanitizers in food processing. Goldenberg and Rafe state that a sanitizer or disinfectant must be efficient under the conditions of use. It must be safe for use by the operatives applying it. It must not influence the flavor or odors of the food processed by the equipment cleaned by its use. It must be easily rinsed, leaving no toxic residue, and it should be easy to use. Next, types of chemical sanitizers. 4 basic types of chemical sanitizers are approved for use in the food industry. These are quaternary ammonium compounds called QARs, iodophores, chlorine-based surfactants, acid ionic surfactants. First, let's begin with chlorine-based sanitizers. Chlorine was first used in Germany to treat water supplies in 1894. In the early 1930s, the use of chlorine solutions to wash and rinse food processing equipment was begun, and by 1946 it was recognized that the use of chlorinated sprays at selected points in food processing lines resulted in lower bacterial counts in the finished product and reduced odors. The major sources of chlorine are sodium hypochlorite, granular calcium hypochlorite, granular chlorocyanurates, gaseous chlorine, chloramine T, and generation from sodium chloride. Of these, sodium hypochlorite is the easiest to use and least expensive. The moiety producing antibacterial activity is hypochlorous acid. This can be formed by any of the following hydrolytic reactions. Chlorine combines with water to give hypochlorous acid and hydrogen and chlorine ions. Similarly, sodium hypochlorite in combination with water again produces hypochlorous acid and sodium and hydroxide ions. In a similar manner, chloramine combines with water to give hypochlorous acid, ammonium ions and hydroxide ions. Hydrogen ion concentration is obviously a very critical factor in determining the hypochlorous acid content and the antibacterial activity that can be obtained from chlorine-containing compounds. pH levels in the 5 to 6 range and below result in the greatest percentage of hypochlorite as undissociated hypochlorous acid, with the strongest influence of pH occurring between 6 and 8. There is currently little confirmed information about the manner in which chlorine is able to exert its effects on the microorganisms. Several proposed mechanisms are destruction of protective bacterial capsules, oxidation of cell protoplasm, formation of nascent oxygen, formation of toxic chloramines, alteration of cell permeability, precipitation of bacterial protein, prevention of enzyme regeneration. Chlorine-based sanitizers normally are employed at concentrations which provide 100 to 200 ppm available chlorine when used as surface sanitizers. In-plant treatment of processed water usually is aimed at producing a concentration from 0 to 0.5 ppm available chlorine, although this varies with the product. Drinking water is usually chlorinated to a level of 0.025 to 2 ppm depending on anticipated contamination of the water and the level of residual organic matter. Contact time and concentration of chlorine are critical factors. At 4 ppm, a 90% reduction in the total count is obtained within 1 minute, whereas with only 1 ppm hypochlorite, the same level of reduction requires 10 minutes. The benefits of chlorination are many. However, it should be emphasized that chlorination does not replace good sanitation and careful management practices, nor does it improve a raw material or finished product of questionable quality. Moving on, iodophores. Iodophores refer to iodine-based sanitizers. If iodine is complexed with a surface active agent such as alkyl phenoxypolyglycol ether, a relatively high concentration of 25 to 30% is solubilized for use as a sanitizer. Such a compound is called an iodophore. The general structure of iodophore is wherein L represents an acyl radical derived from lanolin fatty acid, R is a hydrogen or a C1-4 alkyl group, X is a compatible ion. R2 and R3 each represent a C1-4 alkyl group, R4 is an alkyl, aralkyl, hydroxyalkyl or unsaturated aliphatic hydrocarbon radical. Like chlorine-type germicides, iodophores are active against both gram-positive and gram-negative bacteria as well as yeasts and molds. Most iodophores maintain their activity over a fairly broad pH range, which permits them to be mixed with acidic materials such as phosphoric acid. As a result, iodophores possess not only surfactant and disinfectant properties, but also are capable of removing or preventing the buildup of scale. This property has made these compounds especially useful in dairy industry, where the accumulation of milk stone, that is calcium and magnesium phosphates, is a problem. In this context, iodophores may be used in clean-in-place systems. Iodophores are more costly than chlorine-based sanitizers. However, they normally are used at much lower concentrations of about 12.5 to 25 ppm than the latter. In addition to cost, other disadvantages are corrosiveness, especially on galvanized iron, release of free iodine at temperatures greater than 43 degrees centigrade, and high chemical oxygen demand in plant waste waters. Additionally, certain types of plants which process starch-containing products are unable to utilize iodophores because of the tendency of iodine to form a purple complex with starches. Despite these disadvantages, iodophores remain highly regarded as germicides because they can penetrate organic soil, are unaffected by hard water, and are relatively non-irritating and therefore useful as hand dip for food plant workers. Next, quaternary ammonium sanitizers. These compounds are also called quartz, have wetting properties. However, their principal use in the dairy and food industry is as a sanitizing agent. Their chemical structure can be represented as where R1, R2, R3 and R4 are organic groups such as alkyl, methyl, benzyl and cetylbenzyl. It is thought that quart molecules actually adsorb to the surfaces of the bacteria and so are more efficient on a mole-for-mole basis than most other sanitizers used by the food industry. Their principal action is bactericidal, that is, bacteria are actually killed. However, most gram-positive bacteria are destroyed and gram-negative organisms, many of them significant in foods, may not be appreciably affected. Usually, the growth rate of gram-negative bacteria in quart solutions is relatively slow, and if used within a day or two following dilution from the concentrated quart, there should be no problem with these materials. Quarts are substantive and so provide residual antibacterial activity on treated surfaces. The most common use concentrations are 500 to 1000 ppm. These compounds are stable in presence of organic soil. They are colorless, odorless, non-corrosive and non-irritating. Greatest effectiveness is achieved at a pH level of about 10 and at high temperatures. Now comes the combinational approach. The combination of detergent and a sanitizer into one formulation has the advantage that both cleaning and sanitization procedures can be accomplished simultaneously. In addition, as is the case with quartz and non-ionic detergent, combinations of the two may be more effective than either alone. Proprietary mixtures of non-ionic detergents and quartz as well as anionics plus chlorine-releasing compounds are available from commercial sources. Sequences of detergents and sanitizers may be used to exploit a specific biological circumstance. For example, Wilson and Nelson in the year 1979 studied detergent sanitizer combinations and found that a sequence of an alkaline cleaner to which 50 ppm available chlorine had been added, followed by a rinse and an acidic detergent treatment, seemed to sensitize the spores of an anaerobic organism to a subsequent treatment with an iodophore solution containing 25 ppm available iodine. This combination has produced virtual sterility in food storage tanks used to store tomato products. Next important topic is the antibiotics used in the food. Antibiotics are secondary metabolites produced by microorganisms that inhibit or kill a wide spectrum of other microorganisms. Most of the useful ones are produced by moles and bacteria of the genus Streptomyces. Some antibiotic-like substances are produced by Bacillus species and by some strains of Lactococcus lactis. 3 antibiotics have been investigated extensively as heat adjuncts for canned foods: subtilin, tylosin, and niacin. Niacin, however, is used most widely in cheeses. Chlorotetracycline and oxytetracycline were widely studied for their application to fresh foods, whereas natamycin is employed as a food fungistat. In general, the use of chemical preservatives in foods is not popular among many consumers. The idea of employing antibiotics is even less popular. Some risks may be anticipated from the use of any food additive, but the risks should not outweigh the benefits overall. 15 considerations on the use of antibiotics as food preservatives were noted by Ingram et al., and several of the key ones are summarized. The antibiotic agent should kill, not inhibit the flora, and should ideally decompose into innocuous products or be destroyed on cooking for products that require cooking. The antibiotic should not be inactivated by food components or products of microbial metabolism. The antibiotic should not readily stimulate the appearance of resistant strains. The antibiotic should not be used in foods if used therapeutically or as animal feed additive. A system of classifying bacteriocins that places them into one of the 4 classes has been presented. The Klenhammer system is based primarily on the genetics and biochemistry of these compounds. Class 1 includes the antibiotics such as niacin. Class 2 are small heat-stable peptides such as lactasin F. Class 3 are large heat-labile proteins such as helvetacin J, while Class 4 are proteins that form a complex with other factors. Unlike antibiotics, bacteriocins inhibit only closely related species and strains of gram-positive bacteria. They consist of small proteins and most are plasmid mediated. It appears that some species and strains of all genera of lactic acid bacteria possess the capacity to produce bacteriocins or bacteriocin-like compounds. Although early attention was focused on the lactics associated with dairy products, producing species and strains have been recovered from meat and other non-dairy fermented products. Let's take up individual antibiotics and begin with niacin and other bacteriocins. Niacin is a polypeptide agent that is structurally related to subtilin, but unlike subtilin, it does not contain tryptophan residues. The first use of niacin was by Hurst 51 to prevent the spoilage of Swiss cheese by Clostridium butyricum. This is the most widely used of these compounds for food preservation. It's a hydrophobic compound and it can be degraded by metabisulfate, titanium oxide, and certain proteolytic enzymes. The compound is effective against gram-positive bacteria, primarily spore formers, and is ineffective against fungi and gram-negative bacteria. Among some of its desirable properties as a food preservative are that it is non-toxic, it is produced naturally by Lactococcus lactis strains, it's heat stable and has excellent storage stability. It is destroyed by digestive enzymes, it does not contribute to off flavors or off odors, it has a narrow spectrum of antimicrobial activity. Typical usable levels for niacin are in the range of about 2.5 to 100 ppm. Niacin has been combined with low heat to destroy L-monocytogenes in cold-packed lobster meat. In addition to its use in certain canned foods, niacin is most often employed in dairy products like processed cheeses, condensed milk, pasteurized milk, etc. Its use is also permitted in processed tomato products and canned fruits and vegetables. It's most stable in acidic foods. Because of the effectiveness of niacin in preventing the outgrowth of germinating endospores of Clostridium botulinum and the search to find safe substances that might replace nitrites in processed meats, this agent has been studied as a possible replacement for nitrite. Employed in culture media without added nitrite, the quantity of niacin required for 50% inhibition of Clostridium botulinum type E spores was 1 to 2 ppm, 10 to 20 ppm for type B, and 20 to 40 ppm for type A. The structural genes appear to be the same for niacin, subtilin, and other antibiotics. The cell target for these agents is the cytoplasmic membrane, where they depolarize energized bacterial membranes, that is, reduce the transmembrane potential and form voltage-dependent multistate pores. The result of a pore formation is the loss of accumulated amino acids and the inhibition of amino acid transport. A niacin-resistant mutant of L-monocytogenes has been shown to significantly contain less phospholipids in its membrane. This has led to the assumption that the membrane targets for niacin are phospholipids, which implies that fewer phospholipids would make membrane less susceptible to pore formation. Another important antibiotic is monensin. This antibiotic was approved by the FDA as a cattle feed additive in 1970s and is used primarily to improve feed efficiency in ruminants. Its amino acid sparing action has been demonstrated in fistulated cows. It inhibits gram-positive bacteria and thus its long-term use has the potential of shifting the gastrointestinal tract bacterial biota from one that is normally gram-positive to one that is more gram-negative. Like niacin, monensin is an ionophore that destroys selective permeability of the cell membranes, and the two agents compare favorably as feed additives. Moving on, natamycin is another antibiotic. Natamycin is another antibiotic that is a polyene and is quite effective against yeasts and molds, but not bacteria. Natamycin is the international non-proprietary name as it was isolated from Streptomyces natalensis. In granting the acceptance of natamycin as a food preservative, the Joint Food and Agricultural Organization and the World Health Organization Expert Committee took the following into consideration. It does not affect the bacteria. It stimulates an unusually low level of resistance among fungi. It is rarely involved in cross-resistance among other antifungal polyenes. DNA transfer between fungi does not occur to the extent that it does with some bacteria. The use of netamycin is limited as a clinical agent and it is not used as a feed additive. It has been shown by numerous investigators to be effective against both yeasts and molds. It has been studied that 1 to 25 ppm of netamycin were effective against 16 different fungi, mostly molds. To control fungi on strawberries and raspberries, 50 ppm of nystatin are required for effectiveness. In controlling fungi on salami, the spraying of fresh salami with 0.25% solution has found to be effective. Netamycin appears to act in the same manner as other polyene antibiotics. It binds to membrane sterols and induces distortion of selective membrane permeability. Because bacteria do not possess membrane sterols, their lack of sensitivity to this agent is thus explained. Let's move to another class of antibiotics, that is the tetracyclines. The efficacy of this group of antibiotics in extending the shelf life of refrigerated foods was first established in fish. The tetracyclines are both heat sensitive and storage labile in foods, and these factors were important in their initial acceptance for food use. They are used to treat diseases in humans and animals and are used also in feed supplements. The risks associated with their use as food preservatives in developed countries seems clearly to outweigh the benefits. Subsequently, the effectiveness of chlorotetracycline and oxytetracycline in delaying the bacterial spoilage of not only fish and seafood but poultry, red meats, vegetables, raw milk and other foods. CTC is generally more effective than OTC. The surface treatment of refrigerated meats with 7 to 10 ppm typically results in shelf life extension of at least 3 to 5 days and a shift in ultimate spoilage flora from gram-negative bacteria to yeasts and molds. When CTC is combined with sorbate to delay the spoilage of fish, the combination has been shown to be effective for up to 14 days. Next, Subtilin. Subtilin is an antibiotic which was discovered and developed by the scientists at the Western Regional Laboratory of USDA. It is structurally similar to niacin, although it is produced by some strains of Bacillus subtilis. It's effective against gram-positive bacteria, is stable to the acid, and possesses enough heat resistance to withstand destruction at 121 degrees centigrade for 30 to 60 minutes. Subtilin is effective in canned foods at the levels of 5 to 20 ppm in preventing the outgrowth of germinating endospores. This antibiotic may be just as effective as niacin. Its mode of action is similar to that of niacin. Another important antibiotic is tylosin, which is a non-polyene macrolide, as are the clinically useful antibiotics erythromycin, olandomycin, and others. It is more inhibitory than niacin or subtilin. It has been found to be effective for canned food. When 1 ppm was added to cream-style corn containing flat sour spores and canned, no spoilage of product occurred after 30 days with incubation at 54 degrees centigrade. Tylosin also finds its usage in animal feeds and for treatment of some diseases of poultry. As a macrolide, it's most effective against gram-positive bacteria. It inhibits protein synthesis by associating with the 50S ribosomal subunit and shows at least partial cross-resistance with erythromycin. Dear learners, this was all about today's program. Hope you have learned some new and exciting things. Thank you for your attention. Have a great day ahead. Goodbye.